Good afternoon, everyone, and welcome to today's webinar sponsored by NAVBO's Education Committee. I'm Amber Stratman from Washington University in St. Louis, and I'm going to be moderating today's session. Before we begin, I'd like to remind you that all NAVBO activities are now easily found in the calendar of events on our website. Please be sure to register for some upcoming events. Throughout the presentation, please type your questions in the question box in the side panel. These questions will be answered at the end of the talk. This webinar is being recorded and archived on the NAVBO website for future use. So today I'm pleased to welcome our speaker, Dr. Stefania Nicoli from the Yale University Cardiovascular Research Center. Dr. Nicoli received the 2022 Judah Folkman Award, and today she will share her award lecture entitled RNA-Based Mechanisms Guiding Endothelial Cell Behaviors, which she presented at the IVBM meeting in 2022. To give you some background on the work of Dr. Nicoli's lab, as we all know, cardiovascular disease is the #1 killer in the general adult population in the US and contributes to poor prognosis of many other human conditions such as cancer, neuropathologies, and viral infections. Endothelial cells, as a critical component of the cardiovascular system, adapt to tissue environments by acquiring specialized functions such as the ability to transdifferentiate into hematopoietic stem cells and pattern immune and perivascular cells, such as neuronal stem cells, neural crests, and smooth muscle cells. However, a major challenge in the cardiovascular field has been the inability to understand how these specialized functions are acquired by endothelial cells. This is where Dr. Nicoli's laboratory has come in, and the goal of her work is to understand just that, the mechanisms that endothelial cells use to specialize their function in various tissue environments. During her tenure at Yale, she has placed RNA-based mechanisms of gene expression regulation at the forefront of her studies on cardiovascular function. Her lab has elucidated how post-transcriptional regulation of specific mRNAs is key to endothelial cell specialization, and how this mechanism functions as an environmental rheostat, modulating cardiovascular tissue function in response to different environmental conditions. Ultimately, her work has provided a new way to interpret and predict cardiovascular disease susceptibility and is leading to preventions of ongoing cardiovascular disease. Her work has transformed the understanding of cardiovascular tissue health by helping to identify critical modulators of cell mechanisms that explain endothelial cell homeostasis. So today we'd like to congratulate Dr. Nicoli once again on her discoveries leading to the 2022 Judah Folkman Award, and we're looking forward to your talk.
Great. Thank you very much, Amber, for the kind invitation to repeat this talk, which is I treasure very much, and also for this introduction. Okay, let me just move myself from the screen. Yeah, I would like to start as I started last year with truly an inspiration, and how for me Judah Folkman was essentially my scientific rock star when I was a young graduate student in Laboratorio Marco Presta, as I was studying actually at the time the effect of fibroblast growth factor FGF in angiogenesis, and as you can imagine how much I look up at him and his research to essentially understand the principle of angiogenesis. But I was also inspired by him, by what he himself treasured as a human being. Indeed, one of his most prestigious awards that he received in his career, which there were so many to list, but one of the one that he treasured the most was dedicated to Ellen Keller, which she is the first deaf and blind and woman, human being, to receive a bachelor and a college degree, basically from Harvard. And she truly championed paradigm shift and changing dogma of what a human being can be. And for this reason, I truly think that both this figure and as Judah Folkman encompass the research that try to change and disrupt the current dogma. I felt inspired by these individuals, so that I wanted to present for the entire rest of my talk actually one story from my lab that tried to change some of the important dogma in molecular biology. And for this, I really need to introduce in a very simplistic way what we are studying in the lab, And as many of you are interested in transcription, and therefore the production of messenger mRNA that is then transported in the from the nucleus to the cytoplasmic compartment, where ribosome then synthesize a protein. And therefore, most likely, as we are used to think about to a molecule mRNA correspond a molecular protein. However, we very much know that this is hardly the case, since throughout this very complicated process, there is many mistakes that nature made. And that's why there are other mechanisms, such as, for example, small non-coding microRNA, which try to correct these mistakes, so to speak. Particularly, it seemed like more than 70% of the entire messenger mRNA have microRNA binding site, and therefore, most likely during this pressure, they are, so to speak, excessively transcribed, and therefore are present excessively in the cytoplasm. And that's why microRNA enter in the picture and repress them and balance the amount of mRNA and protein needed for the cell. So we have been studying this process, as Amber mentioned, for over a decade now, and every time we are trying to remove microRNA, and therefore, if you can think about, we kind of release the idea to increase mRNA in the cell, different type of organisms, cell and tissue differentially respond to environment. For example, if you remove microRNA from embryo zebrafish per se, in a population of embryo, they respond differently to stress sensitization and increase their variability to phenotype. If we do similar manipulation in a cell-based approach, we have found that cell actually respond very differently to the extracellular matrix, which is, if we think is actually one of the first environment the cell have to counteract, including the stiffness of the matrix. And finally, we also found that similarly in the context dependent of stem cell in their niche, removing microRNA increase the variability and heterogeneity of the stem cell in the place of birth of their production. So in essence, the overarching narrative through this year of study is that every time we have an aberrant regulation of mRNA, cell become very much sensitized to the environment. So we wanted to tackle the question whether perhaps cytosolic mRNA have in itself an intrinsic ability to sense environment and therefore to help cell to counteract the environment. So the mRNA is actually localized to place where the environment is sensed. It's something that has been interested in discover in multiple contexts. For example, this particular paper highlight how mRNA is localized at the filopodia. Of course, many know Bob Singer lab, the localization of beta-actin, but other mRNA are localized actually in the direction where the cell had to migrate. In this actually was a paper in endothelial cell. There are other mRNA localized in fibroblasts at the lamellopodia per se. And then there are other evidence also in vivo. This is an epithelium in which some type of mRNA are localized in the apical site versus other mRNA localized in the basal sites. And this is in the intestine, it seems to be particularly important for absorption of certain type of the food intake. So clearly mRNA is, rather than be cytoplasmatic in everywhere, seem to have a kind of a smart guess. And whether this is driven by environmental parameter, it's clearly not very understood. So we decided to get to this question by starting to a very simple mechanism, which is the localization of mRNA to focal adhesion. So focal adhesion, as I mentioned, is the sentinel, what I would call it. So of course, there are macromolecular complex of protein. They are at the base of each cell, and they sense the substrate, meaning the extracellular matrix. But they are, in a sense, as you can see by this movie, extremely dynamics, and they constantly are in a state of assembly and disassembly, while the cell continuously explore in a monolayer the environmental cue dictated by the extracellular matrix. So we felt like that perhaps this would be the perfect location to understand whether mRNA processing or mRNA localization, translation, any type of mRNA function may be coupled with the ability to sense environment. And we performed this experiment in which, it was a protocol originally established by the Claire Waterman lab, where it's possible to isolate within a monolayer or of cell focal adhesion via a mechanical and hydrodynamic isolation of the cell body, which enable essentially focal adhesion to be stick with the surface of a plastic. And by doing so, is essentially possible to have cells that look like this, where we remove in this picture right here completely the nuclei as well as part of the majority of the cytoplasmic compartment, and we kept mainly the focal adhesion here depicted in vinculin focal adhesion protein and of course actin, which is a component incredibly attached to the focal adhesion. And this is just a control of the enrichment of focal adhesion of this PrEP versus other type of subcellular compartment. So reassured by the use of this strategy, we then went ahead and then isolate from this focal adhesion isolate all we can know about mRNA, including their polyadenylated sequencing, the protein, as well as the ribosome footprint to understand if there is any mRNA there, where it's translated, where making the protein, what kind of protein they're making, and so on and so on. So the results of this experiment are represented right here. And particularly, we use two cell type, UVEC, which is very familiar for the NAVBO community, but as well as human dermafibroblast. The reason why we wanted to know, generally speaking, two different adherent cell type for conservation purpose for adherent cell. And what strikes us in the beginning of this analysis is that indeed there were enriched mRNA focal adhesion in both cell type, and that's also consistent with other discovery mRNA focal adhesion, including beta-actin. But what was unusual is that the majority of the RNA, nearly 70 to 80%, were actually considered untranslated by the fact that the protein they were encoding, it was not found in focal adhesion. But more importantly, the ribosome footprint actually suggested that they were not covered as much as, for example, the translated mRNA. However, the mRNA per se was highly abundant. Another way that we try to validate this phenomena is by taking one mRNA at a time and performing two assays. One assay, the visualization of the mRNA itself with a single molecule fish right here. And the second assay is called PurePLA, which is an assay that allows you to visualize the native polypeptide. And we noticed that while the translated mRNA, the mRNA and the protein polypeptide were somehow coincidentally near focal adhesion and was quantified right here, the other mRNA that we classify as non-translated and translated were actually decoupled by this correlation, and those are here. And it's important to notice, I'm pretty sure you recognize, for example, beta-catenin and TRAC and NET. So those are all mRNA that are indeed necessary for cells. So they are translated in the cytoplasmic compartment at a certain level, but focal adhesion were found essentially poorly translated. So we also went ahead and tried to visualize them in a more massive way. And for this reason, we use merfish, which is essentially a single molecule fish, but now brought it to all the mRNA that we identify as focal adhesion. And we use the cycle mRNA control, which essentially those were probes that recognize mRNA encoded for cycle protein, which are not were not found enriched at focal adhesion. As you can see, this is a focal adhesion staining as well as a merfish staining were depleted by focal adhesion, however our RNA were indeed nearby and on top of focal adhesion as well. And we can also perform some quite precise quantification of where are these focal adhesion compared to, what are the mRNA compared to focal adhesion, and suggesting that mRNA, about 50% of the mRNA were either on top of focal adhesion, and then the more far away to 1 micron to the focal adhesion were actually being not enriched anymore, while the cell cycle RNA were continuously enriched, suggesting that there is some type of confined behavior whether these mRNA form are nearby a focal adhesion protein complex. Another interesting thing we can do with this data is essentially analyze whether there is a spatial geometry on the distance between the mRNA and the focal adhesion itself. So we apply what usually is a machine learning strategy to actually correlate the mRNA expression in a cell. In this particular, we use a focal adhesion and we take the mRNA nearby and we ask, well, what is the characteristic of focal adhesion based on the mRNA that have nearby? And that resulted essentially in finding 4 types of focal adhesion types that are defined by the type of mRNA nearby. And those are actually, interesting enough, distinguished by their morphology and circularity. And we have other parameters right now. But for example, mRNA that were nearby a focal adhesion state 1 is clearly defining a focal adhesion that is smaller and is also more circular. And the others in state 4, for example, are more elongated and more bigger. And as you may know, focal adhesion size is essentially a readout of the tension that they are applied, and therefore bigger focal adhesion are likely to be in a much tension, and therefore in a mature stage versus a native one, they are smaller. And their mRNA are quite precise in defining the functional state of focal adhesion. For example, here is a 4 of mRNA classified as untranslated, nearby state 4 as well as state 1, and again these are nearby the smaller versus nearby the larger focal adhesion. Interesting, the mRNA that are actually founded as translated, such as tallin and fibulin, which of course they are important for focal adhesion protein, are equally distributed across focal adhesion. What is suggesting therefore that there is something in the sequence of these mRNA independent by the protein translation that seem to be associated with the protein function at focal adhesion. Now, clearly, to understand what is this potential function, we want to go back to the basic and try to understand how can an mRNA molecule regulate protein without necessarily encoding for a protein function and protein signaling. And the viral field is teaching us so much nowadays, including the fact that an mRNA can interact with an RNA binding protein and changing multiple function of the protein itself. But there is also other fascinating concept which are predominantly identifying the nuclear, whether RNA can actually function as a crowding agent via formation of condensate or aggregate, call it in different ways you please, but essentially the goal of this RNA is to keep molecular biochemistry component nearby to a particular reaction, such as in this particular case, transcription. So we felt like in either 2 scenario, what is key to identify is clear the RNA binding protein that is mediating this function. And for this reason, we perform a proteomic assay in which we again remove the body of the cell and keeping the focal adhesion as the substrate. In this particular case, we did it in 2 conditions. One condition with a gentle permeabilization RNases. So we remove RNA so that we essentially release whatever focal adhesion protein or RNA binding protein attached to focal adhesion. And we also use cyclohexamide, which is an inhibitor of translation, as control. And here I show essentially what the results point out quite clearly to us, that the most enriched focal adhesion, RNA binding protein and focal adhesion, particularly G3PP1, but all of these actually are stress granulose protein. And the majority of this protein decrease dramatically after we remove RNA, and they don't necessarily decrease as much as quite not significantly with the translation. So suggesting that those were potentially a good candidate for us to investigate how RNA can be essentially trapped in a protein complex in this way. So for the initial screen, we actually start with G3PP1 mainly because it was the most abundant focal adhesion, and the second reason is because when we do an immunofluorescence staining of this protein in HDF and the fibroblasts, it was obvious that why having a broad cytoplasmatic staining, which is quite normal, I will say, for RNA binding proteins to be so promiscuously expressed. They also have a very specific expression in the lamellopodia, and this particular expression is obviously highly enriched with focal adhesion, where the majority of the very dynamic adhesion are localized. And when we treat this cell with this gentle permeabilization with RNase, we actually remove the localization of this RNA binding protein and focal adhesion. We also identify that this protein was connected to focal adhesion protein. G3PP1 actually can be pulled down if you pull down paxillin, and this type of pull down is this diminished or completely gone when you remove RNase, suggesting that G3PP1 may indeed be the protein clustering those RNA, something that we have validated by using, again, MERFISH in a G3PP1 loss of function cell. And so MERFISH in this particular case was done with shRNA, HDF, and this is the same library of focal adhesion mRNA that we selected in the very beginning. And as you can see, when we remove G3PP1, the distribution of our focal adhesion mRNA is fairly equal to the cell cycle. So they kind of step back to their preferential localization at the focal adhesion. This is also confirmed by this dual localization of both RNA as well as this puro PLA. Besides the quantification, one interesting aspect that we found that whenever we remove G3PP1 and therefore the mRNA is kind of backing away from the focal adhesion, the RNA does not re-enter in translation, suggesting that essentially the localization of mRNA at focal adhesion is not necessarily to pause translation, which is something that's been proposed for other type of mRNA. G3PP1 dynamically interact with focal adhesion. So this RNA binding protein in this movie is depicted in green, and the paxillin is one of the core focal adhesion protein in red. As you can see, the focal adhesion actually does assembly first, and then G3PP1 arrive approximately 45 seconds later, and then it will be matching with paxillin and the assembly as well as the disassembly of focal adhesion for the rest of the lifetime of the focal adhesion per se. So indicating again that this interaction is extremely dynamic with focal adhesion assembly and disassembly. For that reason, we were not surprised that when we removed G3PP1 from the cell, we actually saw a striking diminution of assembly focal adhesion, a striking diminution of disassembly, and a profound diminution of cell migration. Again, this RNA binding protein, it is therefore required functionally for the protein complex to be together and to function during the main function of focal adhesion, so the migration. So why this is encouraging, it did not yet answer or why an RNA still with G3PP1 need to be incorporated in a focal adhesion or in a matter of fact in any protein complex. So for that reason, we dig in the literature about how G3PP1 function. So G3PP1, as I mentioned, it's a stress granule protein. What they do, they form this very spheric complex, and within their complex, they are indeed trapping an mRNA that is depleted by the ribosome. So in essence, every time there is a cytotoxic stress in a cell, and by cytotoxic we mean like very nasty cytotoxic stress, including arsenite, shock, and so forth, there is a stall, so the translation stop, and therefore the cell try to basically capture these increased mRNA. They are now have no purpose if not to be captured and therefore degraded later on. So indeed, the concept of having mRNA in the cytoplasmic compartment is mainly as a product of discard and therefore no functional signaling. So we have focused on understanding what kind of complex these proteins making nearby focal adhesion. And to do so, we have used high resolution microscopy to visualize the type of G3PP1 complex together with the RNA that we identify a focal adhesion. This is just one example of this type of imaging. So you can see three different type of shape and granules right here with one mRNA molecule, in this case TRAC2 with the paxillin. And first of all, we knew that these are ribonuclear complex because when we treat with RNase, again, these complexes are dissolving and therefore you can see this kind of A's type of staining. And the second thing is that clearly the G3PP1 granules that we are seeing in focal adhesion are not similar to stress granules, which are very spheric, but they actually have a variety of shape that goes from very branched and elongated to actually more smaller, but still quite irregular, which is triggering our idea that perhaps the RNA that is incorporating this complex is far more different to what this could be a stress granule type of mRNA. And to address this, we actually took, a tube approach where it's possible to reconstitute the granules as well as the mRNA in a tube. And those are, for example, 4 types of mRNA that were found at focal adhesion and were incorporated with the G3PP1 protein at the concentration which is usually used for stress granule. So again, we confirmed that all our focal adhesion mRNA make irregular granules, and some of them, they were more circular than other, but nevertheless, they're mainly varied in the branch size rather than in their circularity. And that for us was a major clue telling us there is something in the sequence of the mRNA that is dictating a very peculiar shape of these G3PP1 granules. And we wanted to understand what is the sequence that make these granules so different for what we knew about G3PP1 stress granules. We did this by taking 2 mRNA localized at focal adhesion, and again in a completely reconstitute complex assays in a tube. We essentially start chopping 500 base pair starting from the very beginning of the coding sequence to the very end of the UTR, in the hope that we could found the sequence that were making these mRNA in different shape. And what we identify is that indeed, in a completely random matter, at least at the moment we don't understand where localization matter, but so far we found indeed that there were sequences that were more prone to make branched and more other sequences that were more prone to make smaller and irregular granules. And interesting, the sequence of that distinguished the 2 types of granules were not necessarily dictated by the AU ratio, GCR ratio, which is something that was essentially found before in the biology of the complex of condensate, but was indeed the main difference that characterized the different granules of the free energy dimerization. which essentially mean that RNA or the sequence in blue right here, they were essentially more prone to form this circular or spheric granule, are likely to interact with themselves, while the one that formed the more branched type of granule were likely to interact with themselves and forming essentially with multivalent interaction and RNA-RNA interaction. So that really triggered our curiosity here, right? So we started with the idea that we have an mRNA depleted by ribosome, potentially now able to function as some type of mRNA sequence. And now we reduce this concept to essentially a 500 base pair fragment, one that have different property compared other in terms of cell dimerization. So we wanted to understand essentially why do we even bother about coding and non-coding. Let's just take one of these fragments and try to understand what do they do in vivo. The first thing we did is to take this fragment and observe their behavior in the context of cell. And you can do this by essentially stabilize the mRNA by fusing what's called an MS2, a viral sequence in which is gets recognized by another RNA binding protein, MCP, tagged with the GFP. So you can essentially visualize your RNA in green, and then you can do movie, including having focal adhesion like this. And you can study the behavior of each of these RNA at focal adhesion by performing basically turf microscopy. And so you can finally also quantify their speed as well, in particular, whether they are localized in a certain manner. And this is, as you may recognize, is philopodia, where the focal adhesion highlight right here. And the first thing, it was very obvious to us that when these fragments are nearby a focal adhesion, they actually are tend to have a different speed compared when they are in the cytoplasm. Particularly the fragments that are branched had seemed to be a very interesting slow speed compared to the one that were defined as circular, and why this difference were not observed in the cytoplasmic compartment. So the behavior suggests that pausing nearby a focal adhesion, as has been observed for other mRNA that actually are translated, is suggestive of function. And one way to address this is by performing another trick here, in which you tag this fragment of mRNA to a focal adhesion protein. In this particular case, you do this by fusing a focal adhesion protein with the RNA binding domain of this MCP that then will recognize the mRNA and trap it in the protein nearby itself. So in this case, we trap paxillin. Again, this is a normal expression or normal localization of this untranslated mRNA at paxillin, and this is with the trap system. Now we basically overcharge paxillin with this fragment of RNA, which they distinguish by this multivalent interaction. And it was very interesting then to follow essentially what is the behavior of either the focal adhesion and overall the behavior of the cell. Well, the first behavior that we tested was actually migration. And we had tested using, as we did for G3PP1, the actual Wundt-Dillon assay. And we saw some interesting surprise. So first of all, when we linked paxillin to this fragment, they are classified as circular, so they form circular or G3PP1. The cell migrate way much faster compared to the control, as well as when we actually link to paxillin the fragment of multivalent interaction, they actually quite slow compared to the control. So depending on the fragment that we link it to paxillin, we can essentially make cell either migrate much faster or slower. And it's interesting that we also use different parts of the fragments, so not only for TRAC, but for example, for other type of mRNA that were found in focal adhesion. And we can recapitulate this data, and we can actually change quite significantly the speed of the cell. So something that it's really empowering the way like we can change cell migration by changing this RNA composition and focal adhesion. So we took a look at this cell to the focal adhesion assembly at this assembly state, and what it was obvious that both RNA, when they are trapped at focal adhesion, they actually increase the assembly rate. Or whether you have a circular or a branch type of G3PP1 RNA, you actually change quite dramatically the disassembly rate of paxillin. By having particularly the branch type of RNA having a positive impact in the lifetime of focal adhesion, while the circular one have decreased the lifetime, and that's why essentially they migrate faster, and the other one they migrate much slower. So what they actually do to the protein. So what we are currently investigating, that this is all about protein mobility and the ability essentially to interact with each other. We did this first assay in which we frap paxillin, which is a method essentially to understand the mobility fraction of each protein. And as suspected, whenever we link a branch mRNA to paxillin, paxillin have a hard time to move, and therefore their percentage recovery is significantly low compared whenever we link to a circular RNA, which now we increase the protein mobility, and therefore the ability to disassembly is seen to be advantageous. Now, this is again the beginning, and probably we're really scratching the surface of this potentially interesting mechanism in which cytoplasmic mRNA perhaps have alternative motive to be there beside protein translation. We are currently keep investigating the mechanism of function, but so far what we identify is that the ability of this mRNA to self-dimerize change the milieu, potentially the biophysical milieu in which protein are embedded, by therefore facilitating their protein-protein interaction or actually inhibiting their protein-protein interaction based on their mobility. And of course, focal adhesion complex are the perfect setup for this mechanism. Protein mobility is pretty much the core of their mechanistic function, beside, of course, signaling. So with that, I also would like to look at the future. What is I found this extremely exciting. We just essentially found mRNA therapy as at the forefront of the way that we can think about cure disease in the context of cardiovascular disease by many other. And now we can also think about how mRNA can be engineered to change fundamental cell functions such as migration in the context of disease. So with that, I'd like to thank you, the member of the lab. They are actually working in this project and many others related to basically the potential untranslated functional mRNA, particularly Anna Boras right here, which has been the leader of this project, and Laurent and Thornton and Peter Martino. There are two newly graduate students in the lab, which are also jumping into this project. Of course, many collaborators to help us to really dig this process. And finally, everybody in the lab for supporting each other and be a really good team. And happy to take any question.
Wonderful. Thank you so much, Stefania. That was a great talk. Feel free, people, to start typing your questions into the questions tab. And maybe while people are getting their questions typed out, I'll take the honor of offering up the first question. So a lot of your data is linked to focal adhesions. And I agree with you. It seems perfectly logical to signal there. Have you guys tested now if you change the stress on the focal adhesion? Or maybe I missed this. Can you speed up or slow down translation just based off of stiffness?
Yeah. So Yeah, so that's actually something that Lorraine studied. So partially part of these RNA that we found as localized focal adhesion are translated, and the translated protein are indeed very important protein for focal adhesion, including taline, filament, and yada yada. So when actually we perturb the mechanical components, so including stiffness, the translation of this protein does change. So suggesting that there is another side of the project here where translation is regulated by mechanical environment. What we did not yet test is whether the untranslated change in their actually composition or as well localization. And we don't think that the two phenomena are yet connected. So for example, we don't imagine so far that a non-translated mRNA will become translated in certain condition. The reason why I'm thinking is that way because the type of RNA that we found classifies untranslated encode for protein that will not necessarily be useful for the mechanical part of the focal adhesion protein. So that's the current model. However, of course, we need to more extensively prove it.
Yeah, that's super interesting. My other question along these same lines is, there are plenty of cells that are adherent, but there are lots of cells that are not. And do you have a working model for, like how an immune cell might utilize the same system, but in a different way?
Yeah. Yeah, it's an interesting question. The majority, so these RNA are clearly not specific, for transcriptionally speaking, are not specific for adherent cell. They find it most likely in many other cells. So some of them indeed are, structural ER, or so they clearly seem to be in everywhere, suggesting that they might have different property depending of depending on what type of protein-protein interaction a cell might need. It's very hard to say and to speculate because it's so out there at the moment that I could potentially imagine. My line of thought, is fascinating, but if we really, the theory of the RNA as being the first molecule to be actually made, one potentially interesting idea that RNA, because it was able to replicate it without DNA, maybe it was the first one to create actually a structural component for cells to stick together. So interesting enough, it would be very interesting to go evolutionary speaking to go back to the first organisms, they were able to form an adhesion in a mechanical environment and see whether those RNA are evolutionary conserved independent of the protein to do this crowding kind of agent process. So that's where I'm kind of very interested in going to see at the end of the day, what does it make this RNA functional, and think the way like we can now discover maybe new mutation. We are very focused on deleterious mutation coding sequence, perhaps mutation that maybe don't do single nucleotide, that don't do anything on coding, they perhaps destroy this multivalent interaction. So clearly we are thinking ahead of what this property can really bring to the table.
Yeah, and that kind of leads us into our next question, and that's you have such beautiful live imaging, and most of your studies were done, it looked like, on not junctionalized cells. But if you ever caught cells that were like junctionalized to each other, do you ever see transfer of these RNAs between cells?
Yeah, very interesting. I definitely have to ask to Peter that done all these movies, he ever done it. So we were kind of rigorous in keeping the cell not as a model layer, but more sparse, because of the fact that in that condition, focal adhesion are the main driver of the mechanical response. So as soon as you induce cell-cell contact, the whole focal adhesion geometry change quite dramatically by moving them more toward the junction. So therefore, you intrigue a mechanism that would not help us to really study assembly disassembly in the way like we know in the. So that's why at that time, we really were sure not to do that. But now we are, of course, very interested in that we understood much better the mechanism, what happened with cell form a monolayer, and we are currently investigating that.
Awesome. Well, I think we are out of questions in the question box, but if you have any additional questions that you're thinking of, please feel free to reach out to info@navbo.org, and we'll make sure Dr. Nicoli gets your questions. Oh, here we go. One more. Binding between, let's see, binding between mRNA and G3BP1 is via the sugar moieties or the bases?
Yeah, very interesting question. So, technically speaking, for what we understood in the biology, and of course, Paul Taylor, it's Roy Parker are the gurus on G3BP1. What we understood that per se, there is no appropriate RNA binding motif that would justify this so many RNA to actually be in complex with G3BP1. So what the best that we can think about is about the ability to form a proper complex, whether you call them condensate. Not sure if I stretch it that far, but essentially the ability of G3BP1 to actually form a proper, with the IDR domain, to form a proper aggregate and then recruit and concentrate RNA. It's a main property. In a sense, why you need part of RNA binding, it doesn't necessarily mean that the RNA had to have an RNA binding motif for G3BP1, which they don't have, the majority of them. Some they have, but they're not. So how the nucleation of these physiological granule form is yet to be fully established. So we are kind of inspired of how the stress granule form, and that's how we apply some of the principle on that.
Awesome. Okay, so one more here. Is G3BP1 lectinic?
Was that lactinic?
Lectinic, yeah. Lectinic? Teninic?
You can educate me. What is?
Maybe I can unmute Raphael. Here we go.
It's kind. We can wait there if you want to chat in.
There we go. Let's see. Unmute. Okay, Raphael, I've unmuted you if you would like to speak up.
He'll have to unmute his mic as well.
Oh, okay. Bind sugars. He says bind
sugar. I'm interested in it. So why would you? What are you exactly thinking? You clearly have something in mind there.
Yeah, if you want to unmute yourself, Raphael, you can.
Yeah, well, it has to do with the thing that when the G3BP and the mRNA, in the RNA you have the component of sugar, but you also have bases. Question is if they interact through a protein sugar interaction with lectinic, if they interact through recognition of the bases of the RNA and some amino acids in the protein.
Yeah, so again, it doesn't seem that the recognition of the RNA, it's all justifiable by an RNA binding motif. So clearly it's more about the nucleation of the complex through this IDR, this disorder domain into the protein, and then which usually is triggered by binding 1 or 2 RNA, and that's how the IDR open up and that the aggregates start to form. So that's based on stress granule biology. So whether there are sugar modification on the protein, like thinking of glycosylation or not, I actually don't know that particular. It could be actually interesting to look at it. Yes, it might change slightly the function based on where the protein should be localized. Thank you.
All right, unless there are other questions, then we will thank Dr. Nicoli for her talk today. We hope you enjoyed today's webinar. Look out for more information for future events on the NAVBO website. And thank you for joining us today. See you next time.
Thanks.